You didn't even read the title of the talk? Especially for you, right? 12 minutes, 26 slides. Anyways, okay, so this is a little bit of an experiment for a talk because I've modified the talk as I've listened to everybody present, and so I'll take the liberties of being the last talk and try to integrate some ideas based on what has been said so far. So with that in mind, I'll kick it off and I'll go as fast as I can. I will not go over time. So, real title, Understanding Cancer Genomes and Transcriptomes. So I'm going to start by saying, there's been discussion about how you fund, and there will be discussion about how you fund open source software and successful models for developing software for bioinformatics, for instance. I'm going to lead with what we do in the Galaxy project and what I do personally, and I've published a number of papers with the Galaxy teams, but this is certainly my point of view and not necessarily representative of the complete team. In my research, I often do genomics until I can't do genomics anymore. So I use Galaxy, I use command line tools. I find a computing challenge somewhere along the way. Once I find this computing challenge, I try to invent new technology or write new software to address that challenge, and then try to demonstrate the usefulness of this new technology or software by actually going back and doing the genomic analyses. So what this means in practice is that at the end of the day, we have a paper that has both technology and biology, and it's published usually in biotechnology journals, for instance. In the case of what's going on here with cancer genomics and Galaxy, the simple computing challenge, or the broad computing challenge, is analyzing cancer genomes. In particular, I'm going to talk about tools, workflows, and visual analysis that we've added to Galaxy to make this process a little bit easier. And the particular investigation that we're talking about, or that we're looking into, is pancreatic cancer transcriptome analyses. And I have very little time to talk about this today, but you can find me online if you're curious, or offline if you're curious. Okay, so I need a show of hands. Who's actually used Galaxy? Most people? Okay. I'm going to walk through just a little bit about Galaxy to make sure we're all on the same page. I am a member of the Galaxy team, as is Afghan, who's talked a couple times today. Galaxy is an open web-based platform. Accessible, reproducible, and collaborative computational genomics is where we stand. So we hit on accessible, reproducible, and collaborative all the time. These are the core foci of Galaxy. From a user perspective, Galaxy is a GUI for doing high-throughput, high-performance genomics. And so for users, three very important things can be done with Galaxy. You can get your own data, you can integrate with public data, you can analyze data and create complex workflows, and you can do things once you have done analyses such as visualization, sharing, and publication. Galaxy is available on lots of different platforms, as we've heard today. From a developer's perspective, so many of you in the room are developers, Galaxy approaches the world from a modular plugin architecture, where we define generic attributes and behaviors of these modules, and then you can write implementations of those as necessary. Examples in Galaxy include tools, data types, file formats, data sources, so where you're getting your data from, whether it's public repositories or sequencers, data stores, so different file systems, whether it's online or offline or local, job scheduling engines. We now have support for visual analysis and visualizations that you can just plug into Galaxy as well, and I'll mention that as I go through. I'm going to skip the demo, actually. We'll just move on. But most of you that use Galaxy, you understand that you have a web-based platform, you upload your data, you run tools, new data sets are generated, and you do this iteratively to build up complex analysis. Okay, so I'm sure most of you understand that and are aware that cancer genomics is big, right? Cancer genomics is one of the interesting things you can do from a medical genomics perspective because cancer manifests itself in the genome. So we can study it quite well with the current technologies we have. On the top is a discussion of particular cancer tests. They're using next-generation sequencing to assess the aggressiveness of prostate cancer. And at the bottom is a more general discussion of how cancer centers are racing to try to develop the infrastructure, as Francis just mentioned, to try to figure out how to analyze cancer genes more accurately. Okay, so what we've done with Galaxy, new tools. I'm going to talk very little about the new tools that we've put into Galaxy today. I'm going to focus mostly on the workflows and the visual analysis. But the new tools include things such as variant calling, fusion detection, variant annotation and filtering, as well as VCF manipulation. Okay, so here we go. We kick off. Here's a Galaxy workflow. This is a really simple single sample transcriptome analysis if you want to potentially analyze something you get from a tumor. It does things such as gene fusions at the top, identifies gene fusions, does transcription, identifies individual isoforms, computes expression levels, and then goes down and computes variants. Right? Pretty straightforward, but is a necessary building block to do something more advanced. Okay, so here's a slide I put in partway through the talk, or partway through the conference to argue that Galaxy workflows have potential advantages, right? So There are lots of different ways to set up pipelines. They all have their advantages and disadvantages. I think, we've heard today a couple disadvantages about Galaxy workflows. In fact, we've heard they're somewhat brittle and they don't always scale well. We're going to fix those someday, hopefully someday soon. But in the meantime, I want to highlight some of the positives as well. So with Galaxy, it's not a black box, which is very important. So we talked about this notion of recomputability, and with Galaxy, you can swap things out. So if I go back to this workflow here, if I wanted to swap out the aligner, so top hat is right here, top hat 2 in this, but I could swap out star, right? It would take about 4 clicks for me to go and substitute star into this workflow. And I would like it to be easier. I'd like to be able to run multiple alignments and then continue this on. We'll fix that someday in Galaxy, but for now it's possible to swap star in or swap your favorite spliced read aligner into this workflow and continue on and rerun it with minimal challenges. People that don't know how to program can do this, so again, if we're going to try to get at cancer biologists, pathologists, and whatnot, Galaxy presents a potentially useful way to do this. All the stuff at the bottom, what is Galaxy? Galaxy enables you to take this object and do things like import workflows, export workflows, so you can move them from one Galaxy instance to another and run them. You can share them online via static URLs. You can publish them and you can embed them in Galaxy Pages, which are these interactive online supplements that you can write in addition to your papers, where you can include data sets, workflows, and visualizations that you've done, and point people to these resources when you publish your paper. So I would argue these are some nice advantages with Galaxy that are not available potentially in other platforms. Okay, so what can we do? Once we have this initial workflow, we can do things such as Brad talked about. You want to compare your call variants with something, right? So the something we're going to use is something called the Cancer Cell Line Encyclopedia of the Broad Institute. What they've done is they've done partial sequencing of lots of different cancer cell lines. And we have 6 patients here, and we have the cell line data. And the question we want to ask is, how similar is this cell line to the patient? So I want to know whether the cell line is a good model for these patient tumors that we have. This happens to be the pancreatic cancer data that I mentioned before, the pancreatic cancer transcriptomes. And what you can see is that the cell line has more variants in general than these patients do, at least for these subsamples pulled from the Cancer Cell Line Encyclopedia. So OM stands for OncoMap, HP stands for hybrid capture with probes. What's more important, though, is that you see that the cell line looks relatively different in the patients. So in this particular case, this cell line, which happens to be a relatively popular pancreatic cancer cell line, does not look especially like the tumors that we see in patients. Not a good model. Okay, another thing we can do with this is we can go beyond and we can start to augment what pathologists want to do. So in particular with pancreatic cancer, you pull out the tumor, you often want to know how much of the biopsy is actually tumor cells versus how much is normal cells, right? And so what we see here is that this is the information we got back from the pathologist. It looked like the biopsy from patient 4 had no tumor cells in it. At least that was the initial claim from the surgeon. But we went back and we looked at it, we looked at the number of variants, and we compared it. And there's clearly variants there that suggest that there are tumor cells. It's a low percentage. So it's probably in the order of 30%, give or take, given what we know about patients 5 and patient 6. But this is a way to get, or to combine the genomic results with what's already being done in the clinic to provide a better estimation of tumor percentage that they can get just by looking at the sample. Okay, I don't have time to talk about clustering patients via gene expression, but I have a technology track talk tomorrow afternoon if you're curious. So there's some clustering done there. This is the complex workflow I've developed. It takes variants plus gene expression data plus annotations, and it tries to boil it down to say, show me the variants in differentially expressed genes that are potentially deleterious. So these are frame shifts, these are non-synonymous indels. And the upshot of all this is that we can move towards targeted EQTL analysis. So EQTL analysis, for those that aren't familiar with it, is this notion that you can tie mutations in the genome together with gene expression. And so the ability to do this requires you to, again, be able to call variants, be able to find differentially expressed genes and the variants in those genes, and then finally be able to annotate and say, let's throw out all the ones that are synonymous and likely to be benign, and let's keep the ones that are likely to be deleterious. We've actually had good luck with this. With this particular workflow, we've identified some interesting mutations in known genes that have been identified before as being having an impact on pancreatic cancer. So I think we're optimistic about this. Okay, I'm going too slowly. Visual analysis. So here's the traditional analysis that you see in Galaxy. You use tools to operate on data sets. You generate more data sets using tools. What we've done over the past years, we've incorporated notions of visualizations and visual analysis. So we have these visual applications. They can visualize data using the web, of course, where everything is done in a web browser, no data downloads, no software downloads. But we can also tie data sets and tools and visualizations together to do visual analysis. And so what we have with Galaxy is we now have this state-of-the-art infrastructure where we can manage data and pull only the data out that we're looking at one time. So we're doing this over the web. We're not downloading our 50 gigabyte band file, but we want to be able to look at it. And so to do that, we have multi-level caching. We have advanced things, that's a real technical term. We have advanced tools that do automatic indexing of the data sets along the way. And so they can pull this data out both at the aggregate level. So say I want to look across a whole chromosome and look at coverage data. That's totally different than zooming down into a gene level region and pulling out the individual reads. You can share and publish these visualizations. So you can provide a URL once you've created something and said, here, take a look at my visualization, perhaps of this patient data. And quickly send it to a collaborator. And we have this framework that's now made it possible for you to take your web-based visualizations written in Canvas, D3, HTML5, stuff like that, and stick it into Galaxy just like you can put tools into Galaxy. Okay, so I will show one quick demo. Let's see here. So I'm going to, I showed something I think similar like this last year. So here's Galaxy's genome browser. It's yet another genome browser, right? There's a bunch of called variants at the top. We're looking at the DNA repair gene. So DNA repair is known to be relatively important for cancer because if cancer can repair their DNA, cancer is more likely to be aggressive oftentimes. So what we're seeing here is a bunch of variants from our 6 patients. Right now you see the mouse on the far right. It's highlighting a particularly strong set of variants, or at least it appears to be a strong variant. It appears in all 6 patients. So what I want to do though is I want to be able to recall these variants in real time and change my parameters to see if I increase the stringency of the variant calling, how that actually affects the output. So what we're getting at here is we want to combine tools and visualization in real time with large data sets. And so what we'll do here is we opened up VARScan. It's a tool, common variant caller. Interestingly, it uses a frequentist approach rather than a Bayesian approach. It's somewhat of an older variant caller. But it still works relatively well as long as your coverage is sufficient. What we're doing here is we're changing the parameters of VARScan and we're rerunning our variant calling in real time. This took, genome-wide at least, for 6 patients, it took about 2 days to call these variants. Here we'll do it in a matter of minutes, only on this gene, because Galaxy's going to automatically pull out this data we're looking at and then rerun it with VARScan in real time. Now we see by increasing the stringency, we lose a bunch of variants that were not very strong, but we're still left with our one variant that appears quite prominent in our data. We can continue to rerun as much as we want, right? So this is the advantages of this interactive visual analysis. We take large data sets, we scale down into individual gene level regions or multiple regions, and then we can analyze them in real time because Galaxy will pull out the interesting, or the visualized data and use that. Okay. Now, the downsides to all this are that you can only look at one region at a time, and you're kind of shooting in the dark potentially. So this is a tool we developed, and it allows partial parameter space exploration. So we built up this partial parameter tree, and we're going to sweep across different parameter values and look at the variants being called. Okay, so I'm configuring the tree. I'm changing some parameters that have to do with the read coverage as well as the number of reads that show the mutation. Here I've kicked off 4 different runs across 2 DNA repair genes rather than 1 now. So we're looking at ERCC1 and ERCC2. And you'll see the variants come out. Now, this provides a more systematic way to choose your parameter values. So we're after these good parameters, right? We're doing this exploration here. We're trying to see in real time how changing parameter values changes the tool output. Once I decide on parameter values that I like, then I can go and I can rerun it on the whole data set and wait 48 hours until I'm done. This is definitely for exploratory research. We'd love to do this in a machine learning fashion where we have some objective function that we try to get towards, and there's automatic sweeping that goes on in the background. But this is a nice first step to allow people to see how parameter changes impact tool output. I'm almost done. Okay, so what you've seen is interactive use of a real tool in real time to look at real data and call variants in real time on a small region or a small subset of genes. Workflows are coming soon. So we're going to integrate this workflow so you have multiple steps rather than a single tool. Acknowledgements. The Galaxy team is large and they do fantastic work, and I'm really happy to be able to build on top of their work and show you what I was able to show you today. We're generously supported by NHGRI as well as NSF and Emory and Penn State where we're located. My principal collaborator at Emory at the Winship Cancer Institute is Mike Rossi. If you want more information on Galaxy, follow the links on the left. Galaxy project is good, and then we have our code repository is Bitbucket, and then our wiki is at the very bottom. If you're curious about Galaxy in particular, or visualization in general, I have a longer talk about this. It'll be more biology oriented tomorrow. And then, as Nils mentioned, we have an integrated visualization computing workshop on the last day of ISMB as well. Thank you.